Sorry students, links got expired. Suddenly I came back. So join in everyone please. So let everyone join in. So I will start, at least we have 50 students. So please join in. So we have 31 students, now it's 28. Now to start with the next topic, animal cell culture. बच्चे video quality is already 720p. So you can see, so we can have 720p of quality, so it's no problem. So just check your internet settings, how is it, but from my side it's all good. So I will start now. I think we had quite long break now. Animal cell culture. So cell culture in vitro, this is a, will tell you a brief history first of all. So in 1885, rocks maintain embryonic chick cell alive in saline solution for a short length of times. Alexis Carroll cultured connective tissues and showed heart muscles tissue contractility over for 3 to 3 months. Then in 70s, Gordon Sato published the specific growth factors media requirement for many cell types. Then in 80s, tissue culture became less of an experimental research field, a more of widely researched tool actually. So what are the applications of cell culture techniques? It helps to study the cell behavior without the variations that occur in the animal. Artificial organ development, in vitro propagation of virus, vaccine development, drug effect, recombinant DNA technology, all being covered in the cell culture. Characteristics of cells could be maintained. Finally, it avoids legal, moral, and ethical problems of animal experimentations. So there are some limitations of cell culture. Takes times to learn aseptic techniques. Quantity of material is limited. Cost of media, maintenance of cell line is limited in this case. So there are some cultures that we have to discuss: organ culture, cell culture, primary cell culture. So organ culture is a three-dimensional culture of undisaggregated tissues, retaining some or of the features of the tissues in the vivo. And then comes the single cells, no longer organized as the tissues, derived from the dispersed cells taken from the original tissues. Then primary cell cultures, they are derived from the explant, that directly from the animals, usually survive for a finite period of time, involves enzymatic or mechanical disruptions of the tissues. Safety level: substances hazardous to health, substance that can cause cancer carcinogens, substance that can cause damage to the developing fetus, substance that can cause mutations in the genetic material. They have to be avoided while doing so, such as gentamicin, thapsigagin, possible teratogens, hygromycins, possible carcinogens, streptomycins, mutagens. So we have to maintain and not to bring them into our cell culture techniques or maintain our level. So some safety measures have to be maintained. Use of the cell culture areas. The cell culture areas, as any other laboratory, is a working area. Do not bring your friends in with you. Do not eat, drink, or smoke in these areas. Do not use his mobile phone. Do wear a lab coat at all times, whether in the cell culture area or laboratory. Do wear disposable gloves. Make sure that dispose of them in correct way before you leave the area. Do not wear disposable gloves in the corridors or write-up areas. So what are the basic equipments that we need? We need horizontal laminar flow cabinets. These provide a sterile environment for the cell culture but offer no protection to the operator. Then we required filtered air centers cabinets directed to the front. Most sterile cabinets are at the back, and we need centrifuge, human drive cells in that case. Contamination of cell culture has to be maintained. So mycoplasma is mostly the reason for contamination there. So antibiotic resistance microorganisms have to be there in your cell culture, and you have to maintain temperature, pH, and nutrient supply to the cells in a tolerant way. So isolation of cell lines for in vitro culture. You can resect the tissues, cells or tissue cultures in vitro. Then you can do the primary culture, then subculture of making a secondary culture. Then further you do the subculture doing cell lines, cell isolations, immortalizations, successive subculture. Then leading to the three different paths: cloning cell line, transform cell line, or you can have immortalized cell line also, or you can have senescence, that is death of your cells. So we have discussed primary organ culture, secondary culture. So primary culture is the one that is directly derived from the animal tissue or the embryo or the adult, normal or neoplastic, doesn't matter. So cultures either are the tissue explants or single cells. Finite lifespan of time they have, mainly encourage dependent, exhibit contact inhibition. Secondary cultures, they are derived from the primary cell culture, isolated by selections or cloning, becoming a more homogeneous cell population, finite lifespan in vitro, retain differentiated phenotype, maintain anchorage dependent, exhibit contact inhibition. Then types of cell culture in vitro. You have to maintain continuous culture that is derived from the primary or secondary culture, immortalized. They have homogeneous cell populations, loss of anchorage, dependency, contact inhibition, infinite lifespan in vitro, differentiated phenotypes, genetically unstable. So their cell morphologies vary depending upon cell type. So there's fibroblastic, endothelial, epithelial, and neuronal. So these are the four types of your cell culture techniques. So types of tissue culture. So they are primary. We have discussed already. Continuous, finite, and definite. Primary are the one with a normal cell culture with change in their division rate, and continuous is the finite and indefinite. In the finite, we have single types, roughly 30 times of divisions. Infinite, indefinite is that like tumor cells. They keep growing and they never stop. Their growth is never stopped. So we have discussed already primary and continuous culture, and this is the stem cell line, stem culture. So morula blastocyst is made, then inner cell mass is there, then embryonic stem cell culture grows, muscle cells, blood cells, neurons, intestinal cells, pancreatic islet cells, liver cells. Further, so stem cells has ability to self-replicate and also differentiate to multitude of different cell types hidden within the body. So they could go for self-renewal or differentiation. In the self-renewal, they produce pluripotent stem cells. In the differentiation, they could go for mesoderm, endoderm, and ectoderm. Mesoderms are the one which are producing RBCs, skeletal muscle cells, cardiac muscle cells. And whereas in the differentiation, endoderms produce pancreatic cells, lung cells, and thyroid cells. And differentiation produce ectoderms, pigment cells, neuron cells, skin cells. In the hematopoiesis, it's not required, this one. So then there is also in the cell cycle, there is a different stages of growth that you all know: G1 phase, G2 phase, S phase, and M phase. G1 phase is the one where we have checked for the cell size, growth factors, DNA damage, and there is a resting state G0 also. Then S is the DNA synthesis phase. Then G2 phase, check for the cell size, DNA replication. Then comes the resting state between. Then spindle assembly checkpoint is there in the mitotic phase, where chromosome attachment to the spindles is happening. And degree of transformation. So you have immortal, faster growth, anchorage dependent, and tumorigenic mice in mice. So if the growth is so high, that is the tumorigenic. Then there is anchorage dependent growth, and there's faster growth also. So basic materials needed for cell growth. So you need substrate, you need nutrients, you need environment, you need sterility. So in substrate, you need your medium to be plastic coated with the ECM, protein suspension culture. Nutrients should have culture media. Environment should have CO2 temperature, carbon dioxide temperature, 37 degrees Celsius, and humidity. Sterility should include aseptic technique, antibiotics, and antimyotics, mycoplasma tested. Then cell culture environment, basal media maintain pH and osmolarity, provide nutrients and energy source. Components of basal media includes inorganic salts, pH indicators, buffers, glucose. So maintain osmolarity, regulate membrane potential, ions for cell attachment, enzyme cofactors. So the pH indicator includes optimum cell growth approximately pH 7.4, and buffers include bicarbonate. Buffered media requires carbon dioxide atmosphere, HAPS strong chemical buffers like pH 7.2 to 7.6, and glucose is the energy source in that case. Cell culture media, components of basal media, you need keto acids, carbohydrates, vitamins, trace elements. So all these materials are required when you're creating your media of a cell culture. And the cell culture environment includes also supplements like L-glutamine, non-essential amino acids, growth factors, hormones, antibiotics, and antimyotics. Antimycotics, so like penicillin, streptomycins, in growth factors. You can have uptake of amino acids to maintain the differentiations. Non-essential amino acids, they are required for the energy source for protein synthesis, and glutamine is essential amino acids for the energy source used in the protein synthesis. Then cell culture environment in vitro. These are the fetal calf bovine serum. They includes growth factors and hormones, aids cells attachments, binds and neutralize toxins, long history of use. They have disadvantages that they're infectious agents, variable compositions, expensive, and regulatory issues. We have to also heat inactivate them for 56 degrees Celsius for 30 minutes, so that destruction of complement and immunoglobulins and destruction of some viruses into them. So this is how you do your cell culture. Revive the frozen cells first of all, maintaining the cell culture, then subculture, then cryopreservations. So this is how you maintain your cell culture, like this in this Eppendorf things. So this is flask, uh, typical flask looks like this. Then comes the passaging of cells. So why passage? To maintain cells in culture, to increase the cell number for experiments. So check for cofluency of the cells, remove spent medium, wash with PBS, incubate with trypsin EDTA, resuspend in serum containing media, and transfer to the culture flask. So how you do it? Passaging of cells, I show you.
The growth of cells in culture follows a standard pattern. A leg after seeding is followed by a period of exponential growth called the log phase. Cells should be passaged when they cover the plate, or the cell density exceeds the capacity of the medium. Maintaining log phase growth will maximize the number of healthy cells for your experiment. Before retrieving your flasks from the incubator, sterilize the hood and have all required supplies at hand. Examine your culture carefully for signs of contamination or deterioration. Handle the cells with care during transport. For adherent cells, remove the spent medium from the flask using a sterile pipette. Rinse the cells with a balanced salt solution such as DPPS. Make sure to use a salt solution without calcium and magnesium, as these may inhibit your cell dissociation reagent. After rinsing the cells, remove the salt solution. Each time you aspirate liquid off the cells, place the next solution on quickly. Add your cell dissociation reagent to remove cells from the plate. Use just enough solution to cover the cell sheet. Consider using a gentle dissociation reagent, such as Triple Express, to avoid damaging your cells during dissociation. Make sure that the solution completely covers the cells. You may want to tap gently on the plate to help the cells detach. Use a microscope to confirm the cells have released from the flask. They will start to appear round as they release from the substrate, and will move or slide when the flask is tilted. Complete dissociation is necessary. Do not leave the dissociation reagent on too long, especially if you're using a reagent other than triple. You may choose to manually break up lingering clumps by repeatedly pipetting warmed medium over them. A single cell suspension is important to achieve an accurate cell count. If you are using trypsin, the collection medium will need serum, or you'll need to use a trypsin inhibitor to inactivate the trypsin. If you're using triple, inactivation is achieved by dilution alone. No serum is needed. Transfer the cells to a conical tube and centrifuge to remove any residual dissociation reagent. The speed and time of centrifugation will vary based on your cell type. After centrifugation, you should have a well-formed pellet. Remove the medium from the centrifuge tube with a pipette and discard the medium into a waste container. Try not to disturb the pellet. Resuspend the pellet with warm, complete growth medium. Gentle pipetting will disperse the cells to ensure a homogeneous solution of single cells. Remove a small sample for cell counting. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless. Using the microscope, count the total number of cells and the number of dead or blue cells. Based on your cell count, determine how much additional fresh medium to add for optimal seeding density. Add the required medium, mix the cells gently, and pipette the solution into fresh flasks. Cap the vented caps on your flask tightly. If the caps are not vented, cap loosely. This keeps contamination out while allowing adequate gas exchange. Use a north-south, east-west motion to evenly distribute the cells and transport the flasks to the incubator. When passaging suspension cells, you'll begin by removing a small sample from the cell culture flask for counting. You'll follow the same counting procedure for adherent cells using Trypan Blue and either a hemocytometer or the Countess automated cell counter. Based on your cell count, add additional fresh medium to the flasks. Stay within the minimum and maximum volumes. This is important to maintain optimal air exchange and shaking flow. You may need to split the culture into multiple flasks. Cap the flasks appropriately, depending upon whether they are vented or not, and return them to the incubator. Every 3 weeks, gently spin the cells, remove the medium, and replace with completely new medium. This eliminates cell debris and metabolic waste.
That was a quite summary. That was a quite summary of everything that we have discussed so far in the cell culture, and we have discussed so far. So why heap is buffer, not PVS? Mainly because HEPAS is widely used in cell culture because of its better maintaining physiological pH. It maintains uh, physiological pH and changes in the despite in the carbon dioxide concentrations when compared to the bicarbonate buffers, so which is also commonly used in the cell culture. That's the main reason. So what are HeLa cells? So I don't remember the full form. They are from the human HeLa cells, the lines belonging to the strain that continuously cultures since 1950 compared to the other HeLa cell only to survive. HeLa cells are immortals because they will divide again and again and again. Expression of overactive telomerase. So they keep dividing these HeLa cells. Yeah, this is an immortal cell line. So that was about passaging. Then if you are done with the passaging, then you do the cryopreservation, in which you resuspend the cells in the serum containing media, centrifuge, aspirate as supernatant, then you mix them in the 10% DMS, so in FCS solution. This is, and then transfer to the cryovalve, freeze at minus 80 degrees Celsius, transfer to the liquid nitrogen storage tank. Then this is the manual count cells. This is how you manually count. In a hematocytometer, you put your cells on the one side and you check in the crosses and check the average of cell number, dilution factor of cell suspension, and then you can calculate your total cell suspension at the end. So cellometer automated lets you view cell morphology for usual visual confirmations after cell counting. take advantage of 300 cells types, save sample images with results securely on your computer. So idle growth curve for cells, this is the idle growth. You have a lag phase, then stationary phase, then comes a stable plateau phase, exponential after the exponential phase, then comes a declining phase afterwards. That's how the normal. So a cell culture contaminated can be defined as some elements in the culture that is undesirable, leads to the unwanted things. So we are done with this part. So these are the common cell lines. Human cell lines are MCF, HL60, HEX, HeLa cells, uh, Henrietta lacks, leukemia, human embryonic kidney cells. Primate cell lines are Vero cos7, another CHO from hamsters, SF9, SF21 from insect cells. So this is an organ culture. These are artificial organs growing in the chambers. So that's it. So we are done with this part. So some images now I will show you some from this animal cell culture. Shall we continue, students, or do you want break, or we do them tomorrow? What do you say? Because I think it will be too much for you. Shall we continue this for today, or we can also continue them tomorrow? Tell me. We do it accordingly. What is the difference between HeLa cell culture and which are some other cell cultures? Their maintenance has to be there for a long time, and they will not grow for longer time, like Huvik cells and all these things. We can do it. They have specific lifeline, but HeLa cells, they keep growing on because of the telomerase activity. Yeah, so we do tomorrow everything. So I know it's too much for you today. So we do the rest of the part tomorrow. How are different cell lines identified from mammalian cell cultures? How are the different cell lines identified from mammalian cell cultures? So I will share you this part. Passaging of cell culture. Some books I am sending you. So I will send you the tissue culture tomorrow. For the moment, we are done for today. I know it's a lot for you today to grasp it. Just go through the slides once more, and then we take care of it. Okay, thank you. Wish you all good night. We maintain this period, like from we start at 8. If we are finished by 9:30 or something, we done with that, so that we can have more energy for tomorrow also. So we try to manage things like that. But today was a lot for all of us. Yeah, it's fine if your assignment is getting exceeded to the 2 page. It's not, I'm not so harsh on you to be like it has to be like that. So I just give you one idea that a summary should not be more than 2 pages of anything. So if it is getting exceeded, it's okay, because sometimes we are learning a lot, we are going through many things, and if it is extending to 3 page or 4 page, it's fine. It's not bad, so bad doing so. So, on that note, thank you so much. See you tomorrow, same time, same place, with more exciting lectures and videos and animations and virtual labs and many things. Okay, then, good night. Bye bye. Take care. Vielen Dank for heute. Liebling student, and it's sehr dir Zum Tag, zum Morgen. Tada, bye bye.